Hello everyone again. So we have 9 people participating. So guys, when Asen was your instructor, what was the policy on attendance?
It was mandatory.
Mandatory. Okay, so people just don't know that it is mandatory, right?
Yes.
Well, so if it was mandatory, I'm going to start punishment for those who don't participate. And I guess we have 16 people here. Okay, so we start with the lecture, and it is about Alamar Blue. And Alamar Blue is the special dye that could get reduced and change their parameter. So the parameter is color first, and second, the fluorescence. So when it changes from blue to pink, it becomes highly fluorescent molecule. Sorry. Okay, so let me turn these things off. Okay, cool. So we start with the general knowledge that we should recap before we start with the cell proliferation assays. So first of all, of course, what is proliferation? Just to understand how it works and why would you want to know about it and measure it. Okay, so as you know, the cells divide. And if we talk about the horizontal division, in this case, we have 2 identical cells coming from mother cell. And of course, if the cell, let's say, gets some damage, so this particular cell got lots of ROS inside, and in this case, the cell undergoes death. So there are 2 possible ways to undergo death. So it's necrosis, and the second one is apoptosis. So this necrosis is the non-controlled manner of dying. And in this case, lots of internal proteins, they get released, and some of these proteins, they become toxins for the neighboring cells. And of course, necrosis is not something that the cell would want to undergo. If it knows that it is damaged, it undergoes apoptosis. So there are 2 possible ways. So first is when the cell decides that it needs to die, and the apoptotic pathway starts. The other way is that the cell is actually receives the signal from the outside, usually T killers. So they send the signal to the cell, and the cell undergoes apoptosis. So in this case, of course, it's a controlled death, and we have the visualization of the cell, and then endocytotic recycle of the compartments. So these are 2 steps that we could, these are two ways that we could choose for the cell. And of course, the other cells that are not damaged, they just keep dividing. And as you all know, the telomeres, the chromosomes, they have these telomeres that actually reduce with every division. And at some point, they are so short that the cells, they become obsolete. And usually for human body, we have about 50 divisions. So if you think that it is not much, you have to understand one thing, that 50 divisions means that one single cell gave rise to 2 to the power of 50 cells. And this number is actually huge. And because we have this number here, we know that the capabilities of our organism is about 150 years. Well, of course, it's related to many things like the possibility to work without any faults, etc., for our heart, for our lungs, for the brain, etc. So, but if we average everything, the 150 years should be reachable for the person if they have healthy conditions and they don't have all those toxic environment, etc. Okay, so this everything is related to the cell division. Okay, so, and of course it's a healthy cell division. Now let's say that the cell at one division, it obtains some trait, and in this case it's a trait of non-controlled division. Let's say the key player of apoptotic way does not work in this particular cell because it has the mutation in this gene. So if we receive the apoptotic signal from the T killer, this cell cannot respond to this particular signal because the key player of apoptosis in this cell does not work. And of course, if it wants to undergo the same apoptosis by its own, so it sends the internal signal, of course, again, the key player does not work, and this actually creates a problem called non-controlled cell. So we cannot control it, we cannot kill it. And at some point, because we have the non-controlled proliferation after we receive this benefit, we have more and more mutations, and the accumulation of mutation usually leads to the overwhelming growth of this particular clone. So these cells divide, creates lots of species of the cell that are different to each other by mutations. So let's say that this particular cell, it has the mutation that allows infinite proliferation, and this does not have it. So this particular one, it will die after 50 division. Of course, it will create a huge amount of cells, but it will die eventually. But if we, in this cell, have unlocked the non-natural behavior of telomerase, this cell will exceed this threshold of 50 divisions, and it will lead to the prevalence of this particular clone in the growth in the tumor. And of course, it will lead to huge problems for the host. So the parasite here, the tumor cells, they will kill the patient eventually. Okay, so what are the conditions for the proliferation itself? So it is related to both of these types of the cells, and it actually is related to any type of the living organism on the earth. So we, first, of course, we need the nutrients. Without nutrients, the cell might go to, so let's say this is a cell, and we don't have nutrients. It goes into the dorm state. Dorm state in this case is quiescent. It does not divide, it does not really leave, it just stalls all processes until we reach the point when the nutrients are accessible again. So it kind of sleeps, okay? So this is happening when we have nutrients or gradual, let's say, decrease of pH, temperature, and oxygen concentration. Not only oxygen, actually, any type of the small molecules that are diffused towards our cell. So if we have the reduction or increase, and main thing here is gradual decrease, then the cell can undergo this process of dorming itself. So it just stalls all processes and tries to live in this condition. Let's say that the change was very steep. So it was living at temperature 37, and then it dropped to, let's say, 65. And of course, this very fast transition between these two steps might kill the cell. So the cell is boiled, and we don't have time to go into a stalled condition. So this everything might happen, and this actually how we tweak our cells. I mean, by tweaking, I mean how we can actually store the cells. So you know that we freeze them, and the frozen cell can be held in the freezer for years. And you know that if you want to transport the embryos, for example, you could do that while they are in the liquid nitrogen. So this liquid nitrogen has the minus 196 Celsius degree temperature, and by gradual freezing of the cells, we could keep the embryos, and as you can understand, embryos are developed organism. If you take just the usual cell culture, it's a monoculture with the clones of the same ancestor. In the case of embryos, they have lots of different cell types that some of them could die in different conditions, but we can effectively keep them in liquid nitrogen for years. And of course, if you want to have a good example, the example could be the Interstellar movie, wherein they were trying to transfer a huge bank of embryos to another planet. And of course, it's achievable with the technologies that we have today. Okay, so now when we know about the proliferation and what it is, so the increase in number, not in size, but in number, okay? So it is called proliferation, and you want to understand how the proliferation happens in this particular cell type that you are using for your experiment. So let's say you are using fibroblasts, and you should expect it to be behaving differently in terms of proliferation to the cancerous cells. So fibroblasts, again, they might divide once in 18 to 24 hours. So usual, very usual for fibroblasts to have such huge number of hours to just divide one cell into two. If you take HeLa, it might divide in 1 hour. So it really differs from cell to cell. I mean, HeLa, they are very diverse now. And some HeLa cells, they can divide in 1 hour, others in 3 hours. So you want to determine what is the doubling time for this particular cell type. And this is why you are doing the cell proliferation assay. Okay, so what is division, which is the basis for the proliferation? So we have division, and the cell usually, so let's say we have cell here, it starts after the previous division when the mother cell divided into two daughter cells. It enters the G1 phase, and G1 means growth 1 or first growth period. So on this G1, it can go quiescent, so it can kind of start sleeping, fall asleep. And most of stem cells, they are in G0 because they don't want to spend anything that they have just to make sure that in the critical moments of injury, let's say, or some bad conditions for the body, they will wake up and produce the needed amount of cells to repair the damage. Okay, so if the cell does not switch to the G0, which is zero growth, and of course you understand that it means stalling everything. Okay, so it proceeds to the next phase, and this is S phase. S phase means synthesis. In this case, we synthesize all the nucleic acids. In this case, both RNA and DNA, because RNA will be used for the production of needed proteins for the G2 phase and the mitosis itself. When we have supercondensed DNA, we cannot transcribe it. That's why all RNA should be accumulated in previous two steps, so G1 and S phase. Okay, so S phase is the replication state. Okay, so by the time of G2, the cell is almost ready with the division, and everything that we need is specific mitotic proteins to actually perform all those actions. Okay, so this whole stage, G1, S, and G2, it is called interphase. Inter means between, the phase between the division. Okay, so when the mitosis starts, we already have supercondensed chromosomes, and these chromosomes, they are inside of our nucleus. And the prophase starts with the disintegration of the nuclear envelope. So the next two steps, prometaphase and metaphase, they are related to the formation of two centrosomes on both sides of both poles of the cell, and the localization of the nucleic content, I mean the DNA content, on the equator of this cell. Of course, tubules start forming by the end of metaphase, and in anaphase, we start pulling these two sister chromatids or sister chromosomes into the poles of the cell. Okay, so the anaphase is moving of chromosomes. So telophase is when both of the DNA copies, they are already in the poles. Okay, so let's draw it like this. But they don't have the nuclear envelope. So telophase is the very short period of time when the cell actually forms this nuclear envelope for two newly developed nucleic acids or sets of chromosomes. Also, at this point, we have the traction of needed proteins, needed contents of the cytoplasm into two directions. So here we have two different types of division. So first one is horizontal, and horizontal means that the mother cell is identical to both of the daughter cells. Okay, so what it means that all content of cytoplasm is divided equally between these two parts. So if you have protein 1, the concentration of protein 1 on the right side would be equal to the concentration of protein on the other side. Okay, so this is horizontal division. But we also have so-called vertical division in this case. So mother cell, and usually the vertical division is the trait or characteristic of the stem cells. So in this case, stem cell, it divides unequally. So one part becomes the daughter stem cell, and the other part becomes the differentiated cell. In this case, if the stem cell is the pluripotent stem cell, then the differentiated cell would become the progenitor, so multipotent stem cell. And in this case, of course, they will not be identical to each other. So both of the daughter cells, they will not be identical to each other. Well, how we do that? We do it by traction of different cytoplasmic content into different poles. So the stem cell or mother cell would be identical to one of them, and the other one would have proteins that are specific to some kind of tissue. So let's say it's a progenitor of neural tissues. Of course, it will have a specific concentration of the factors or proteins that will kind of push this particular cell into the differentiation to the neural tissues. And as you can understand, this progenitor, it will then divide both horizontally and vertically to have different cell types. And when we talk about this vertical division, we mean differentiation. When we talk about this horizontal division, we talk about the increase in number of the same type of the cell, okay? So when we have all the stuff divided between these two parts of the cell, the telophase stops and we start the cytokinesis. And this is the division of the cytoplasm and the biomembrane of the cell. So here you can see a very beautiful picture showing these two cells being separated from each other. So these are two daughter cells of the previous mother cell. And then, of course, everything starts again and again. So that's why it's called cell cycle. And you should know what happens on what period of time, because most of the time you would be using a very specific cell type. And let's say that you are studying the messenger RNA, particular messenger RNA that only is being transcribed in the G1. Of course, you need to know how your cell divides, because you could catch these messenger RNA only during this particular phase. So if you know the cell cycle, you know your work. Okay, so here we are talking about the formation of all tissues and the organisms. So let's say we start with the gametes. So we have one spermatozoon and one oocyte. And in this case, when they fuse their nuclei, they form zygote. And the zygote is the totipotent stem cell. Totipotent means that it can create both embryonic stem cells and the supporting cells. Supporting cells are related to placenta, to the amniotic bag, etc., etc. So something that is not related to the organism or to the embryo. Okay, so if you take these embryonic stem cells, they are pluripotent. Pluri in this case means that it can give rise to any other type of the mature organism. So these pluripotent stem cells, they produce multipotent stem cells, and multipotent stem cells, they only give rise to a very narrow range of the cells that are related to the same tissue. So in this case, the multipotent stem cells, they are progenitors. And if you take these ectoderm, mesoderm, and endoderm, the multipotent progenitors, they can give rise to epithelial tissues, to neural tissues, etc. And the same goes for mesoderm and endoderm. But the ectodermic progenitor cannot give rise to the parenchymal cells because it is not pluripotent, it is multipotent. So there are different levels of potency for the cells. So what is cool about the pluripotent stem cells is that we can actually collect somatic cells from the person and reprogram it. So we give them, so let's say we have fibroblasts here, and usually fibroblasts are actually the ones that are used for the induction. Why? Because they are very redundant and not much mutation happen in fibroblasts. So what we do, we give the special factors, and these factors are related to embryonic state of the cells. So these factors are widely found in the embryogenesis of the organism. So we have special 4 factors, and we add them. In this case, we erase the state of these fibroblasts, and it converts into the stem cell. So after these erase or reprogram, we could give another set of factors. So let's say factors 6 to 7, and push this kind of newly prepared stem cell into one of these progenitors. Of course, there are different progenitors and different factors that are used to push this stem cell into, and we could do that. Okay, so finally, by adding different factors on each step of the formation or differentiation of the stem cell, we can create the cells that are forming the parenchyma of the liver. And you already heard many news about growing organs outside of the body. And this is related to this method. They usually use iPS cells, but with a new law regulation, they could actually use the embryonic stem cells that are obtained from the person on their birth. So let's say that people at age 20 now, I guess 20 years ago it was already done. So the birth command or birth team, they would collect the embryonic stem cells, and then they will put it in the liquid nitrogen. Okay, so they conserve person's embryonic stem cells for the future usage. If the person does not have these embryonic stem cells in the bank, they could undergo the IPS cells. So I means induced pluripotent stem cells. In this case, we have this induction from fibroblast towards stem cell, pluripotent stem cell. Okay, so why don't we use this IPS so much, like everywhere? Because when we use the factors, it is almost impossible to control every single cell so that they don't obtain the benefits or advantages that will lead to the formation of the cancerous cells. So if one of these IPS obtains too much of these factors, it can undergo the transformation. So transdifferentiation from fibroblast to pluripotent stem cell is the very usual process that happens, process that happens in our body. So the formation of cancerous cells is the same. So let's say the fibroblasts, they transdifferentiate into some cells that are not specific to our body. And these cells, we, just because of the tradition, we call them cancer cells. And if we go deeper, the cancer pluripotent stem cells or cancer stem cells. So this transdifferentiation that is not controlled by us, we usually call it transformation. And transformed cells are cancerous cells. So, of course, when we add these factors to the bulk of all these somatic cells, some of them will undergo transformation and they will become cancerous pluripotent stem cells. And we need to filter those malignant cells. And usually, I'm not really sure about it now, but like 5 years ago or 6 years ago, it was a very high chance that together with the IPS cells, you will inject the cancerous cells to the body. So the quality check control should be done many times on the IPS cells, not to kind of infect the person with the cancerous pluripotent stem cells. Okay, so this is the drawback that I'm not really sure if it's already overcome, but 6 years ago it was a problem. Maybe now it's not a huge problem. Okay, so now let's talk about the Alamar Blue. So Alamar Blue is the reagent, and it is related. I mean, the transition from one state to another. So the oxidized one is resazurin, and the reduced one is resarufin. Okay, so these two states, they are related to the reduction of this molecule. And this reduction only happens in the presence of the live cells. Why? Because live cells, they breathe, and the breathing includes the conversion of the specific molecules that can reduce this molecule. So we lose this oxygen here and change it to the hydrogen. And this transition between these two steps is highly dependent on the metabolic activity of the cells. It is very sensitive, and this is one of the advantages of this method. And we can go with a resolution of 50 cells. If we have 50 cells in the solution, we will see the change of the color and fluorescence for these particular wells. Okay, so as I said before, we have two changes in these molecules. So first of all, it's colorimetric. So the change of color from blue to pink. And second, from low fluorescence to high fluorescence. Okay, so these are two steps that you should understand. Just a second, guys. Okay, so we have, we know that it is related to metabolic activity, so we could measure the proliferation is first. Proliferation. Okay, so it's first that we could do. Second, we could check if some kind of treatment is toxic for our cells. So we could measure cytotoxicity. And finally, because this process of the cell breathing is actually related to the any cell type, then of course we could use it to determine if we have some kind of contamination in the liquids that should not contain or should contain bacteria and yeast in very low conditions. So if we have the rise of bacteria or yeast in our milk, of course, it means that we have the acidification of the milk, and we cannot just use it in the production of usual dairy industry. Okay, so you want, if you work in this dairy industry, you want to monitor the contamination. And okay, so about this cytotoxicity, so how it's done. So we have the pharma company. Okay, so this pharma company is developing a drug, and the drug is developed on the basis of many other molecules. So let's say we have 1,000 molecules to be tested, and we know that their formula is actually somehow, let's say, curing some kind of disease. Okay, but of course, many of these types of molecules, they will be non-beneficial or harmful for our cells. So what we do, we take this plate. So let's say it's 384 plate, and of course we need 3 of them to cover only one cell type and all those 1,000 molecules. So what we do, we seed our cells, and then we take time points and measure our fluorescence. So let's say we take only the first molecule out of these 1,000, and we build a plot. So we measure every hour and see how the cells behave. So here we would have the number of cells. And of course, we plot it. So we know that we were seeding, let's say, 10,000. And if the graph just goes like this, of course, at this point, we have the cell death, or even this point. And in this case, of course, we know that, okay, so this particular drug, which is the first, is not okay just because it starts killing cells after 3 hours after addition. Okay, so for some of these graphs, you will have just a straight line. And of course, it means that it is okay for this particular cell type. But then when you filter all those drugs, so let's say you have only 20 left out of these 1,000, and these 20 should be tested on different cell types. So let's say you were using this cancerous cell in this first stage. And this cancerous cell, let's say, the one that we use actually, PC3, it was okay with this drug and no death was shown. But after usage on epithelial tissues, let's say colon tissue, colon epithelial tissue, you see this gradual death of the cell. Of course, you cannot use it because most of the times you deliver the drug through the mouth, peros. And peroral delivery will lead to the contact of this particular drug with the epithelial tissue. So in this case, of course, you measure the cytotoxicity and at some point you filter all the drugs. That's why the production of pharmacological molecules is very expensive, because we have all those checkpoints to pass. Otherwise, this drug will not be approved. Okay, so now let's talk about this small table. So here is shown the ability of different molecules to reduce our Alamar Blue. So as you can see here, it's a usual reaction. So we have O2 plus 2H2, and we obtain H2O. But what we measure here, we measure the potential of these, or energy that is needed to convert these things into H2O, and also to the reduction of the molecule. Okay, so for example, for H2O, we have this E0 is the molar extinction coefficient and is measured in multiple volts. No, millivolts, sorry. Okay, so the coefficient for water is plus 800. So water will not be able to reduce our Alamar Blue because its coefficient is bigger than the coefficient of Alamar Blue. If we take cytochromes, some of them will, some of them won't, because it is very close to our coefficient of Alamar Blue. But if we take any other of these, so the molecules that are related to the cell respiration, they all have this low coefficient, and they will be able to convert or to reduce the Alamar Blue into reduced state. MTT is also a reagent used to count the metabolic activity of the cells. And these guys, they are related to cytochromes and the electron transport chain. Okay, so sorry. All those guys, they are present in all cells, all living cells. And of course, because they are present, they will reduce our Alamar Blue. And of course, it will be shown by the change of the color and the fluorescence in our solution. Okay, so why would you want to use it? And of course, it is because you have a high sensitivity of this method. So when we talk about this high sensitivity, we talk about the resolution of 50 cells for Alamar Blue. If you use MTT, it has resolution about 1,000 cells. So if we have 800 cells in the solution, it actually might not be affected so strong that we see this difference in the color. Okay, but for our Alamar Blue, only 50 cells is enough to start changing the color, developing the color. Okay, so Alamar Blue is super simple and very rapid. If you take MTT, you have steps of reduction. Then you have step of crystallization. So the reduced version of MTT sediments as crystals. Then you need to remove the medium, so aspirate the medium, and dilute this crystal or dissolve this crystal in DMSO. Of course, it means that you kill the cells, but you obtain a very good picture of your cell behavior in terms of metabolic activity. If you talk Alamar Blue, you just go with the addition of this Alamar Blue to the cell medium, and you monitor the changes of color after 1 hour, 2 hours, etc. So it's rapid, simple. It is flexible because you could use it on any type of organism. Again, the reduction happens because we use the reduction by the molecules that are related to the respiration. So any type of bacteria, yeast, fungi, archaea, mammalian cells, any like bird cells, etc. So it is safe because it's not toxic. So cells do not obtain any harm from this particular reagent. Of course, it's safe for lab personnel. It does not interfere with the electron transport chain. Okay, so if you want to use the same cells after you've done the cell proliferation assay, you could do that because, again, it is safe. So about the plan for the experiment. So we have 96 well plate, and these 96 well plate has rows and columns. So every 3 column we have replicates, and these replicates, since they are 3, they call triplicates. Okay, so in this case we have 4 times triplicates, 4 sets of triplicates, but in real life you might have triplicates of different treatment. So let's say group 2, 3, and 4 are drugs 1, 2, and 3. Okay, so you would have different conditions, etc. Or if you don't have different conditions, you could use all those groups to monitor your outliers, etc. Okay, so the rows here, they represent different concentrations of cells or different density, seeding densities. Okay, so in this case, we double every time, but in the last we give 100 instead of 80. I don't know why it happened. I will ask Ajan. Okay, so what do we have here to explain? Actually, nothing. Okay, so we switch to the next step. So you want to work in the biosafety cabinet or under the hood in the sterile conditions, because the cells should be kept in the incubator, and you don't want to contaminate your incubator. That's why you use everything, you perform everything under the hood. Okay, so you add 20 microliters per well, and this is 10% of the final volume of the culture. And we are talking to Aygirim Kushurova. Aygirim, are you here?
Yes.
Okay, tell me, what is the final volume here? That's first. And second, what is the volume of medium?
So
Very well, very well.
So that 10% will be cells and 90% will be DMF?
Look, you add Alamar Blue 20 microliters per well, and these 20 microliters are 10%.
So, like, what is the question? I didn't understand.
Okay, so the question is, what is the final volume here, or volume of the well, if 20 microliters is 10%?
200 micro.
And what is the rest, or DMM with cells? Again. Just speak out, because if you make a mistake, it's okay. No worries.
Isn't it like about the dish size? What is the maximum volume?
No. In the same well. In the same well. So you have final 200, and you have 20 microliters of Alamar Blue. What is the volume of the cells with DMM?
180 microliters.
Alihan and Ayjan, you are, how do you say it? You are warned. That means that I might apply some kind of punishment for you too, because I'm not talking to you now. I'm really sorry, but I'm talking to Egirim. Alihan and Ayjan, did you understand? Okay, so the thing here, guys, don't worry. Of course, I'm not going to apply this punishment, but it is not, I mean, the question itself is not about the right answer. It is about trying to find some weak points of your knowledge or weak points of your understanding. It's not about the right answer, the correct answer. Please stop perceiving it as the competition. No, we don't compete with each other here. We try to teach and learn, okay? I hope everyone understands. If I'm talking to Aygrim, it means that I'm talking to Aygrim. When I talk to Ayjan or Alihan, of course, you will have time to explain stuff, etc. But respect others, respect your peers. So in this case, of course, respect Aygrim, because she is giving the answer. Okay, so Aygrim, you said 180. This is absolutely right. Please do solve the tasks from the internet. Try finding lab math tasks. Watch the videos. I already sent you some videos, so if you don't like my way of explanation, just find one that you like. Learn lab math, because guys, you are third year and your majors is in biology. It means that you should not have any problems with this simple math that is learned on the first year of the studies, because it's again related to your work in the real lab. So why did you choose biology? Because you want to become a researcher, and researcher without lab math cannot work in the modern lab. I would say that today the level of knowledge for the newcomers in the lab is really, really high. By the third year, you should know how to program in Python, in R. And for these two, because you have a very powerful instrument, you would be using these two instruments to do your statistics. And because we have a very fast development of the skills, etc., you will be using data science and machine learning by the end of this decade. So you will have to know Python and other programming languages to apply data science and machine learning to your research. And if you think about the statistics and machine learning, and then think about lab math, you should feel ashamed that on third year, you don't know the lab math, okay? I see it from your assignments, etc., that it is a weak point of your studies. So please understand one thing. I already know the lab math. I don't need it much, but I want you to become a very powerful member of the scientific community. So please learn it make sure that you solve lots of these tasks, because when you solve them in your brain, you have neural networks, okay? And these neural networks, when you don't know how to approach the problem, these neural networks, they form chaotically, okay? So let's say this particular neuron forms connections with other 4 neurons, okay? And one of these connections will lead to the solution of the lab math task. And of course, the next time, it will not form all those connections again and again. It will go directly to the one that was already used. So the network between these two is already formed. And the more you solve the task, the easier it is to go for the signal to go from one neuron to another. And it forms an automated path. So you won't spend much time with the practice. If you don't practice, you will not be able to determine which neuron should talk to which neuron, okay? This is on you. This is your responsibility, because you are the one who wants to get to the lab. It's not me pushing you to the lab, right? So if you really want to become a scientist, do the stuff that scientists do. And in this case, of course, you will form all those neural networks, and it will help you, not me. So again, solve the tasks. And from now on, try to learn all those statistics, etc., because it's not really hard to do, okay? So we coming back to the lecture. So you add these 20 microliters of Alamar Blue, and it's only 10% of your final volume. So you go and check your fluorescence or absorbance. Doesn't matter, because you could use 600 nanometers for the colorimetry. And in this case, you still get very sensitive results for your research, for your proliferation assay. But of course, measurement of fluorescence gives you a much higher resolution than the spectrophotometry. Okay, so after 1 hour, you also take one picture and measure the values, as well as second, third, and 20th hour. So in the end, you will have lots of data that you need to process, and these data will look like this. So you have all those fluorescence intensities for each of the wells, and you already know that you have the triplicates. Also, you know that you have rows, and because you could correlate all of those values, and you could plot the linear regression or whichever linear plot, you could understand how the cells behave in your well. And after that, I mean, after the proliferation assay, you can start planning your experiments with high precision. Okay, so this is the point of this whole cell proliferation assay. Okay, so what you do, first, of course, you want to confirm that variance of your values in all those wells is close to each other. So for the same row in the triplicate, these should be very close to each other. And you look at these variability or variance, and you determine if you could work with this particular plate. If, let's say, in the row A, for triplicates set 1, you have 1, 9, and 10. Of course, it means that one of those is really the outlier. So this variability here, or variance, would be very, very wide. So that's how you try to apply statistics or statistical methods to identify the outlier and, of course, exclude it from your calculations. So what you do, you take the mean, sorry, you take the mean, and in this case, mean, so let's calculate this guy. So it's 20, 1 plus 9 plus 10, so 1, 9, 10, and you divide it by 3. So 20 by 3 is 6, 6, 6, 6, okay? And you call it RFU. So it's not the last, it's not the value that you're going to plot, because you have to blank it or normalize it. So the thing here is that you were having 0 cells in one of the rows. In this particular case, we had a row, not seeding cells at all, because you want to know the background. So the background is actually related to the low fluorescence of Alamar Blue oxidized, and also the plastic wear, etc. So everything can add up to the background. So you want to remove it by using this negative control, okay? So what you do the same calculations for your A row, and you call them blank. And then you correct your RFU against these blank. So you subtract the value of the blank from the RFU, and you get the corrected version. And these corrected versions should be used in the plotting of the linear graphs. So for the cell densities, you are using x-axis, and for the corrected RFU, you are using y-axis. This is how you actually do the plotting. So you could also use the number of hours for x-axis and corrected RFU for your y-axis. So here are different types. So in this experiment, people were looking at different cell types and how they behave in terms of their doubling size, doubling numbers, etc. So for example, you could see that this yellow graph, or human prostate cancer cell line, is very slow on division, while the A549 is very fast dividing cell line. So here you are seeing the plot that shows you the rates of division. So if you look at the first hour, let's say, and see one of the points here, you can drop it on your y-axis. Sorry, you drop it on the y-axis and see how others behave, and at what cell density they actually hit the same value of these y-axis. And then by applying the correlation between the value here, the hour, and the density, you determine the proliferation rate for this particular cell line. So this graph is built on the same cell line. This graph is built on different cell lines. So it means that we could use assay data for different types of processing or calculation. In this case, of course, range in cell densities that you want to understand how they behave in the incubation for this particular time point or time period. And second, you would want to know the maximal incubation time before your graph actually reaches the plateau. So the plateau is actually at 90 or so percentage of the reduction of Alamar Blue, which means that for this particular line here, you obtain the 4 hours reaching 80% of your graph, and it's still okay to use in the experiment. While if you use higher densities of seeding and they reach the plateau very, very fast, and you cannot use it after a particular time of incubation. So I hope you understood what I was saying here. So you don't want to use the cells that are on plateau, because it means they either stall their metabolism, or they compete with each other, killing each other, and the ones that survive, or the ones that win the competition, they divide, others die. So you don't want to use such cells. You want to use cells that are actually showing the growth, okay? And usually it's 70 to 80% of confluency. And this is one of the methods to understand what timing and what seeding should be done for your particular experiment. Okay, so next slide. Okay, so here you could see the experiment from the video, and this second triplicate, so 1, 2, 3, 4, 5, 6, 7, etc. So these guys were excluded from your calculation. I'm not really sure what is the reason. I need to watch the video first, but you are going to do your calculations on datasets 1, 2, and 3, which are occupying columns 1 to 3, and then 7 to 12, okay? So these are three data sets that you are going to use in your lab book. Okay, so again, as the reminder, so we are using different concentrations of the cells, or different cell densities, seeding densities, and then we start doing our calculations. So first of all, we take the A row, and you should remember that it's blank of your experiment. So you find the mean first, and mean is equal to A plus B plus C divided by 3. So this is your mean. Let's call it M, okay? After you identify the mean, you need to identify the standard deviation for this particular data set. So what is standard deviation here? It is the square root from your variance. Variance in this case is equal to A minus M. So A value minus mean squared plus B minus M squared plus C minus M squared, and divided by 3. So this is going to be your variance. And if we take the square root of it, we obtain standard deviation. And it is needed to identify a special coefficient of variance. Usually it's written like this. And this coefficient of variance would be equal to standard deviation divided by mean. And this value should not exceed 10%. If it is exceeding 10%, then you most probably have some outlier here. Okay, so why would you want to have 3 replicates of 3 replicates? So in total, 9 wells for the same condition. It is because when you have 3 replicates, so let's say you have 0.5, 0.6, and 0.8. Okay, so it does not look suspicious now. But if we have 2 sets of replicates more, you might actually see the difference. So let's say this is our set 1, set 2, and set 3. Okay, so here you have 61 and 60. Of course, when you look at other triplicates, you would find that this guy is actually looking like an outlier. And you're going to perform some kind of calculations that will tell you that this is actually an outlier. So you want to exclude outliers. Why would you want to do that? Just to make sure that your experiment is precise. Okay, if you have high value of this coefficient of variance, it means that there might be some problem with the outlier. And if you plot it, so 6, 8, okay, and now you have the distribution of your data points like this. And of course, it's clearly seen that this guy is actually outlier. Okay, so that's why you want different sets of triplicates. And of course, the A row is your blank. So after the blank, after the definition of the blank, you are going to use corrected RFU. Corrected RFU and plot the corrected one. Okay, so here you can see, so for this particular data set, we have standard deviation divided by mean times 100%. It gives you 3.5%. And this is below 10%, which is okay. It means that the distribution of your data points is very close to each other. I mean, is very narrow and the points are close to each other. If you have 10 to 20, you should try to find the outlier because this variance might be actually alarming. So for some kind of research, like drug research, etc., these 10 to 20% might actually lead to lots of complications in the usage of the drug. Okay, so here is what the graph should look like. And as you can see, we have seeding densities on the x-axis and the RFU on the y-axis. I'm going to ask Aijan why they only use 20 hours measurement. So the best here would be having first hour, then second hour, and then 20. I'm not really sure why it happened. Might be related to the loss of measurement. So sometimes we have wipes of the HDD on our computers, and it might have happened at that time. But 20 hours, I will ask her, okay? So I cannot tell why we don't have these first and second hours. Okay, so what you do in your lab book, you are going to measure every data set here and see how these values behave, okay? So then you're gonna plot all of those 3 data sets and see if actually you see some kind of outliers in the graph. So by looking with naked eye here, and here, I would say that for the row H, you actually have the data set number 2 having outliers. So if you look at these 2 data sets only, the mean is very close to 95 or 93. And this guy is already 13 points away from the mean. This guy is even more, like 22 points away. So this might be a real outlier for the whole condition like this. But if you calculate the data first, the data set 2 first, you will see that it's actually falling into a 10% coefficient of variance, which means that you might operate with this value. But after you do the combination of all 3, you might see that this guy is suspicious. Okay, so you should do that. I mean, calculations, you should show it in your lab books. I want to see the calculation of standard deviations. I want to see the corrected RFU for every data set here. And I want to see 3 different graphs in your lab book, okay?
Sorry, can I ask a question about the
Yes,
lab book?
of course.
So since it is really tedious to do all the equations in the Word document, can we use like software to display the calculations like R or something?
You could use R. Sorry. You could use R, you could use Excel. In Excel, it's calculated automatically. So I want to see the formula that you used. And first of all, of course, if you do the handwriting in your lab book, of course, you need to show all the calculations, etc. Not like it was done in the dataset one, and we follow the same logics. No, when you handwrite it, of course, you need to show all the calculations, etc. But now, because of the limitation of online education, etc., okay, just show me how you've done it, and it would be enough.
Okay.
Nice. Any other questions?
Do you mean screenshots or what?
It might be screenshots. You can show the table and the formula in this table. So how you approached it. You really need to control if the values are actually relative and not absolute, because if you use absolute, so in Excel, it's done by $sign. So these absolute values, they usually should not be used unless they are related to some kind of non-changing value, like blank. Okay, so you have to show that you actually know about such values, and you have a proper calculation, assuming that you have both relative and the absolute values. So I want to see it. I want to see that you are controlling everything, okay? So Ayjan, in quiz number 2, you will have all the topics after the midterm. So these are cell light Tallinn and the plasmic reticulum. Which one we had on the last lecture?
So I think lecture 8 was the last one in the midterm about mitochondria staining. And after that, everything will be on tomorrow's quiz, yes?
Yeah, so I just want to confirm which ones. I can't find my. Okay. Yeah. Okay, so we have liposome-based transfection. This is one. Then we have cell light Tallinn, endoplasmic reticulum, and imaging of cells. So these all will be included in our quiz number 2. So again, you will have you will have 15 questions, which means 30 minutes for the attempt. The time corridor is 9 to 9, so 9 a.m. till 9 p.m. At any time you will be joining. So I hope that you, okay, so Labbook 2 must be 100. So Ali, the best would be doing it electronic version, because we are hunting for the plagiarism. So we already had 2 people doing this type of cheating. So plagiarism is not tolerated on our program. So for any type of your work, remember the person who gives the material and the person who copies the material, both are punished. So there is no excuses, there is nothing. If you plagiarize, you both get the penalty. So it's not handwritten, it should be done electronically.
I'm sorry, so today's lecture will not be included in tomorrow's quiz. Yeah?
No, no.
Okay.
It surely goes to your final, but not here. Other questions? Okay, I guess we could say goodbye till next week.
Last question.
Yes.
Sorry.
Go ahead.
Okay. Can you explain again about SD standard deviation? And should we do like for all groups? I mean, 1 to 13 or 1 to 9? Can you open the table?
Yes. Which one?
1 to 12. Yeah. So.
Yes. So for these three, you are going to perform the same calculations. So first of all, you identify the blank, and for the blank and for every other row, you will first find mean, then you find standard deviation, then you show the coefficient of variance, and by showing, I mean you tell if the coefficient is lower than 10% or higher. So the lower one should be fine to use to build your graph. If it's bigger than 10, then of course you might want to see if there are outliers in your dataset. Okay, so after you find blank, you do the correction for every RFU for each row. And of course, after you've done with the first dataset, you're going to perform the same calculations on dataset 2 and dataset 3. Is it clear?
Yeah, but when we plot in the graph, what values should we use, like mean values?
So let's do like this. So you put, again, in your graph, you will have 3 different linear plots, and one goes for dataset 2, dataset 1, and dataset 3. Okay? Is it clear, Ali?
Not so much.
Okay, so for just build plot for every dataset you have. Is it clear?
So like enter each figure into the graph.
What do you mean figure?
I mean like 15.7008, 14.7.
No. As I said, you find the mean. Okay,
So
you find the mean here.
values. Yeah.
Yes. No, not all the mean values. You find the corrected RFU. In this case, you find the mean of the corrections. Okay, so here, first of all, you find the blank.
Okay, yeah, I got it now.
Okay, nice. So
Blank
you find the
will
blank.
be equal to 0, and the corrected values will be much smaller, right?
Well, the thing here is not like this. So you have blank. Okay, so in this case, blank would be 15, almost 15. Okay, so what you do the correction for every single data point in these rows. So for this guy, you have corrected RFU equal to 16.88 minus 15 as your blank. This is your blank, okay? And instead of 16,808, you write 1,808. Here you write 1989144. Here you have 1, etc. And from these guys, now you're finding your mean, and you put it in the table. Okay. So your blank is the background that you exclude from the from your calculations.
And blank shouldn't go into graph, right?
Of course, because in your graph, 15 minus 15 would be equal to 0.
Mhm. Okay, thank you.
You're welcome. Do we have any other questions? Okay, so if we don't have any questions, you still can email me about the stuff you don't understand. Tomorrow, I wish you good luck, and I will see you next week with the Alamar Blue second part. See you guys.
Bye.
Goodbye.
Bye.